I'd like to start with lecture tonight, and we'll do the quiz. I think we'll do it right after the break. The epigenetic inheritance systems part is pretty short. It's true, I didn't have a review sheet. Um, do we need a review sheet? No. I'm happy to count the quiz. You thought the you thought the heritability quiz was next week? Ah, sigh. Okay, it's a really short quiz. I'm happy to bump it over till next week. Um, but sigh. Um, let me take a look. Um, okay, I can do I can do heritability quiz next week. Like I said, it's really short. It's 6 questions. Um, and what I would highly recommend is reading the Ned Block article as well as just going over my lecture. But sure, I'll post study questions and we can do the quiz. We can do the quiz next week. Heritability will not change in content, nor will it be easier or harder to understand for next week. Okay. Okay. Yeah, it'd be really useful for you to have a review sheet. I'm so sorry. I'll post it. I'll post one after lecture. So what are we going to do? We're going to talk about epigenetic inheritance systems, and then we're going to be talking about sociobiology. Mostly will be all multiple choice. Right now, the one that I uploaded was all multiple choice or multiple answer, right? So that's more than one is correct. About 15 minutes, 6 questions, 15 minutes. But if I have another week to puzzle over it, because it's me, I will probably make a couple little changes. So I'm not sure. But right now it's 6 questions, mix of multiple choice and multiple answer. But depending on how quick it is to grade the first quiz, I might stick in a short answer. Then I'd give you more time. I don't think I want to give more time. So it'll probably stick to multiple choice and multiple answer. So then I'll be talking about sociobiology. I uploaded that chapter from the Lalonde and Brown book called Sense and Nonsense. And I'll be talking more in terms of animal examples than human examples, because sociobiology has been much more effective at explaining and understanding social behavior in animals. Because one theme that we're going to be developing throughout this course is that animals and plants cooperate almost exclusively with close relatives. Humans are very different than non-human animals in that we cooperate on very large scales with complete strangers. But we're going to talk about the basically nepotism in the animal world. Okay, so I'm gonna start with epigenetics. And so there's been a tremendous amount of research on epigenetic inheritance over the last decade, although some of the simplest insights have been sort of understood much earlier, but they were called things like maternal effects. Um, the interesting thing now is that we're able to understand sometimes the molecular mechanisms behind traits that are inherited, not due to changes in DNA sequence. So how is that? Well, there can be, well, there are modifications in transcription, how genes are expressed. This is something that in some ways we always knew. In other words, our bodies are composed of highly differentiated cells that are all identical in DNA, right? That wasn't what people expected before they understood how genetics genes are organized. It might have made sense to send, skin cell genes to the cells that are going to become skin cells, and muscle cell genes to the cells that are going to become muscles. And that's just, as it turns out, that's not how it's done. The way it's done is that there are modifications that regulate transcription, how often DNA is read and then translated into proteins, and that shapes the different kinds of cells that we have in our bodies. And so what's interesting is that some of those mechanisms for basically turning on and turning off different genes, many of them are stripped before, over the course of reproduction by making germ cells, eggs and sperm, especially eggs. But some of them are not, especially in eggs. And so the question about epigenetic marking and epigenetic inheritance is, what is its role in adaptive evolution, right? So that's a big question that is still unresolved. So most of the examples I'm going to talk about in this really short review are going to be examples from like disease or failure to develop properly. But like I'm going to say at the end of this little thing, most of our original studies of the effects of genes were on, of huge mutations and, gene, genetic effects that caused either disease or loss of function as well. Okay, so what are some of the mechanisms of regulating gene expression? So there's regulatory modification. We're going to talk about that. That's when like a gene product affects transcription. Structural inheritance, we see this in protists a lot, in like patterns of cilia. Chromatin marking, and so this includes attaching methyl groups. Many of heavy methylation makes it harder to in a promoter, makes it harder for a gene to start being transcribed. Histone modification, which will change how tightly folded DNA is and how sort of visible or apparent it is, accessible it is to transcription enzymes. And then I'm going to talk a little bit about hormonal effects on development in birds and mammals. Okay, so one example, and there aren't a ton of examples, at least that I was able to find, of epigenetic inheritance systems, is self-sustaining regulatory modification. So it's when a product positively regulates that gene. In other words, the product of the gene also causes that gene to be transcribed and expressed more often. And so this is a positive feedback loop, and it will lead sometimes to, sort of stable change in conformation. And one of the best examples of this is the regulator of opaque versus white cell form in a fungus, a yeast, Candida albicans. So the most common sort of morphology is the white cell morphology. They also have it's actually their sexual phase is the opaque cell. So this is a longer, differently shaped, darker cell. It's also one that can conjugate with other opaque cells and combine genes, undergo meiosis, and produce, daughter cells that are the product of that recombination, right? So that's their sexual reproduction. So here we have the two forms. Here's the white yeast cell and the opaque form. And one thing and they make different colors on the plates. And one thing that's really interesting about this system is that there appear to be um, there appear to be um, some immune defense um, benefits to being opaque. In other words, opaque cells are less likely to be recognized by some host immune responses. And so it may also be adaptive to switch to the opaque form. Although I wasn't able to find out what are the costs of being opaque. It's probably they replicate and divide much more slowly. And it's interesting that the two effects of being opaque are somewhat complementary. In other words, what do you want to do if you're trying to evade a host defense, immune defense? Well, one thing is try to evolve rapidly because some of your offspring might be able to overcome that host defense. And that's going to happen when you're reproducing sexually. So it might be that opaque is a better, is a better form to be in when you're under attack by a host immune system. But I don't know what the benefits are of what the trade-offs might be for the white form. But these are two fairly stable states. In other words, it's reversible, but stable. So the product of the gene, WOR1, is a transcriptional regulator that will, positively increase its own production, and so you'll stay opaque. But some cells that emerge from budding or sexual reproduction will revert back to the white form, and then that'll increase in frequency. Okay, another epigenetic inheritance mechanism is structural inheritance. And so there are really just two examples that I know of. One is in the formation of cilia, the patterns of cilia distribution in some protists. It's been studied in ciliates that are, have a lot of cilia. And there was nice sort of early microscopic studies showing, what the pattern of cilia distribution was. What Beeson and Sonneborn did was they actually like physically manipulated the distribution of cilia on the cell surface, and I think they removed some. And what they found was that the cilia itself was a template for the pattern of cilia formation in daughter cells. So basically, when they manipulated the distribution of cilia, that manipulation lasted several generations in the cell cultures that they were studying, which is kind of interesting. The sort of much more ecologically relevant example of structural inheritance is in disease. So there are a number of related brain diseases that are found in humans and non-human animals. In humans, you have Creutzfeldt-Jakob disease, Kuru. In deer, you have chronic wasting disease. In sheep, I think you have scrapie. And I don't know how many of you are actually even old enough to remember mad cow disease. It sort of shut down beef production in England, and there's a good deal of concern about it. And there still is. But what's interesting is these neurological diseases are not caused by DNA or RNA, but by proteins, specifically prions, which stands for proteinaceous infectious particles. So these are misshapen proteins that can cause the same proteins in other cells to also form in the same change of shape. Now, the disease has been sort of studied. I don't. It's not the exact same, but it's also caused by a prion. Its inheritance was studied because there was an outbreak of this neurological disease, Kuru, that was found in the Fore people in New Guinea. But what was really weird was that when you studied pedigrees, one of the things you're going to look at is if there's vertical transmission, that strongly suggests a genetic component to the disease. But if there's horizontal transmission, that kind of disproves a genetic mode of transition, right? So people would study the pedigrees. So you could think of that. So if you study pedigree, you'll see, well, it's actually for humans, we don't do it like that. We'll do it like this. And then you look at children. And so you might find the disease in, more children than you'd expect on average, but then you'd also find it in, unrelated individuals, which means it had to have been transmitted horizontally. That means from one person to another directly, not through patterns of parent and ancestry, parents and children. Now, however, the mode of transmission was eventually shown experimentally, and was shown, and the mode of horizontal transmission turned out to be through cannibalism. So the mourning ritual of this people involved the consumption of relatives, and that could include relatives by by marriage, not just by ancestry and descent. And it took a good deal of sort of work to try to explain to show people why this practice was so harmful, and actually lead to community deciding to change the mourning customs. So Creutzfeldt-Jakob Kuru, there's no adaptive function that anyone can see. There isn't even like a positive trade-off or anything. However, in yeast, like I was saying, was there's multiple indications that the different forms that are being controlled by prions might be adaptive. So in the yeast example, um, one um, one of the prions that was identified um, prevents transcription of a cell wall protein. When that protein is misfolded, then cells produce thicker cell walls. And that will give them a higher survival in environments that stress cell walls, where having a thick cell wall is advantageous. And like I said before, the opaque form is resistant to some immune responses in hosts. So there's good indication that that it can be adaptive in some contexts. So this is another experiment showing adaptive functions of protein misfolding. So what this is using is Benamil, which is a fungicide. Fungicides, the unnatural enemy of fungi. And what this study found was that the wild type, or the more common version, is more susceptible to this fungicide Benamil. But the misfolded protein allows them to still survive even when being exposed to Benamil. Okay, the epigenetic inheritance mechanism that's getting the most attention these days are different kinds of chromatin marking. And this can be either, attachment of molecules like methyl groups to the DNA. It could also be changes to the histones that will alter how tightly folded DNA is, and therefore how easy it is to be read. Heavily methylated DNA generally not transcribed. And what's interesting is that some methyl groups are stripped when forming gametes, but there are others that are heritable. So the methyl groups that you have before cell division and before gamete formation are sometimes persevere into the next generation. So one example is the glucocorticoid receptor promoter in rat hippocampus. And this is a good system because obviously rats are, lab rats. And there's a known maternal effect of maternal care, licking and grooming, that appears to have a non-genetic but heritable component effect on the on a whole suite of behaviors in the pups and as adults. So it will affect their anxiety, their stress hormones, and their own licking and grooming behavior. So maternal care, greater licking and grooming behavior of the mom towards the pups reduces stress and fear of new environments, right? So anxiety and neophobia versus high licking and grooming, which leads to bolder and neophilic um, uh, willingness to explore new environment behaviors. And this is because low licking and grooming apparently leads to methylation of the promoter region for the glucocorticoid receptor. So one of the nice things is that you can study this with, well, we'll see in the next slide, with fostered pups versus their own pups. We don't even need to get to that. So if in low licking and grooming moms, they're the promoter of the glucocorticoid receptor is heavily methylated. That prevents the glucocorticoid receptor from being expressed. The brain has fewer receptors, and that leads to a higher stressed phenotype, higher stress hormones, greater fear of new environments, and lower licking and grooming behavior. So that trait ends up being passed on for multiple generations. And to really see whether this is a heritable trait separate from genes, although there are also genetic effects on anxiety and neophobia. Those are traits you can do selection on. You do a cross-fostering experiment. So you take mothers that are either high licking and grooming or low licking and grooming, and you give them pups, low licking and grooming. And you give them you cross-foster their litters. So you let some moms keep their own pups. And so if you are a high licker and groomer, and then your pups will also, as adults, be low anxiety and high licking and grooming as moms. If you are a low licker and groomer, then your pups will have high anxiety, high neophobia, and will also perform less licking and grooming. However, if you take the pups from a low licking and grooming mom and give them to a high licking and grooming foster mother, these pups will develop the low anxiety phenotype as adults. Interestingly, if you take pups from a high licking and grooming mom and cross foster them to a low licking and grooming mom, they can actually maintain the low licking and grooming, which suggests that something that's going on in the uterus can provide some kind of buffer. So high licking and grooming can kind of rescue the low anxiety adult phenotype, but high licking and grooming also makes the pups more resilient to their maternal neglect after being born. Kind of interesting. Okay, another example of heritable epigenetic inheritance is the effects of exposure to a fungicide. One that's been studied is vinclozolin, which leads to changes in behavior that last 3 generations in rats. So vinclozolin has acute effects on on the individuals that are exposed, and heritable sex specific traits. So males show accelerated onset of adult diseases such as cancer, prostate disease, kidney disease, and I think both males and females show changes in neophilia, neophobia. That's what we're going to see right now. So this is an example of behavioral effect that is tested 3 generations after exposure to this fungicide vinclozolin. So what you have here, what you have in the bar graphs in A is sort of the quantification of the behaviors that you're sort of seeing in B. So I forget how these rats were stressed. I think it was by keeping them in isolation for a few days. But here on the top row, you have kind of a heat map of where they spend in an enclosure when you release them into an enclosure and then sort of videotape them where they are. What you can see is the non-stressed controls actually spend a lot of time in the corners. So what's the right? Get a nice color that'll stand out, and it's not that. Okay. So they're spending some more time in the corners, and when they're stressed, they spend more equal time. They don't seem to have a preferred corner, and they're spending more time in the center. In contrast, and you can see that here, this is outside corner time goes down in controls, but outside corner time goes up in vinclozolin exposed and then tested here 3 generations later. So basically, yeah, they're hiding out in the corner. I don't know exactly what the behavioral significance of this trait is, because non-stress controls also hang out in the corners. I think what the implication is that non-stress controls, when you isolate them for a bit, are going to be exploratory and try to find other individuals, and vinclozolin, morphology rats are going to respond to stress by hiding even more. So another somewhat heritable environmental effect actually is the uterine environment in mammals. So this was studied by Betty McGuire and then a bunch of other folks. And one of the things that she found, one of the earliest results, was that the dispersal distance of pups in prairie dogs, ground squirrels, was affected by the sex ratio of the litter. And so especially females that were born in a male-biased litter dispersed farther, settled and tried to breed farther from where they were born than pups from female biased litters. This is really interesting, bunch of reasons. And as it's been studied further and imaging's gotten better, it's now clear that actually it's not just the sex ratio of the entire litter, but more precisely, it's what's the sex of the developing embryos on either side of you while you're developing in the ovary. So these are, ground squirrels, like pigs, like a lot of mammals, they have large litters, right? So you can have 12 pups developing at the same time. And so if you look in the uterus, here's a female who is surrounded by um, females on both sides. So this would be considered a 2F female. Let's see here. So the female's flanked by 2 females. Whereas this is a 1 male. Male, it's a male that's flanked by 1 male and 1 female, right? And um, the sex of the pups on either side will have effects on behavior and even the sex ratio of the offspring. And because of that, you're going to get some inheritance of these effects because um, a female that's surrounded by 2 males is more likely to have both disperse farther and produce litters that are more male biased. So, prenatal hormones exposure vary with uterine position and the sex of the pups developing, because each pup, like as it's developing, produces sex hormones that diffuse out into the uterine environment and will also impact surrounding siblings. So 2M females, females that have males adjacent to them on both sides, have themselves have higher levels of circulating testosterone. They're more aggressive. They disperse farther before settling. They're more responsive to testosterone, and they're more likely to produce male-biased litters. So there'll be more 2M offspring, which sort of increases that. In contrast, females surrounded by female embryos are going to have more female-biased offspring, more female-biased litters. So these effects end up being somewhat inherited. The system that was studied a lot in the 80s, 90s, and into the 2000s, but not as much. I haven't read as much this decade. Are the effects of yolk androgens in birds. So one thing that was discovered, first described by yes. So 2F females are more philopatric, which means disperse less before settling to use. So this was something that Schwabel described first in, I'm pretty sure this example is looking at zebra finches. And the pattern was that later eggs, so you can see here on the x-axis, we have egg sequence. So it's always going from first to last. And these birds often lay, 4 or 5 or 6 egg clutches. That the last couple eggs usually had higher levels of testosterone. And this is before they developed. So it's not being produced by the chick, it's being I don't know what you'd say, inserted, deposited, um, uh, so this is a maternal effect. It's being deposited by the mom. She is producing these these hormones, the testosterone that's in the eggs, and so she's like controlling the hormone levels in the eggs, and this has a bunch of effects. Chicks with higher testosterone, um, that had higher yolk testosterone before they started development, were more active in begging. And the idea was that this was a way that the moms were helping to even the playing field. Earlier laid eggs um, have uh, have an advantage because they start developing a little earlier. They're going to be a little they might hatch a little earlier and be a little larger. Um, the later developing eggs were given sort of more testosterone to help them sort of beg more and compensate in growth. However, um, that was the original thinking. However, what happened was when they studied um, yolk androgens in a bunch more species, what they found were different patterns in different species. So what we're going to see is sometimes some species, the earliest eggs have more testosterone, some the later eggs have more testosterone, and the behavioral effects of yolk androgens vary by species. So this is kind of weird and interesting. It was studied in house finches, which are a really interesting species for many reasons. They've become kind of a model bird species, partly because it's for two main reasons. One, because um, they used to be native to the Southwest and they were introduced on the East Coast in the 60s. And so they had a big population explosion, range expansion, sort of invasion. And so it's been interesting to sort of study sort of rapid evolution in new environments. And then also because a guy named Jeff Hill noticed that females prefer males with redder bibs. There's a lot of color variation. This is kind of the extreme from least red to reddest. But so this is like range of variation. These are both males from least to most. Lots of red. Females prefer to mate with redder males. One of the first things that he showed was that bib coloration is in some ways an environmentally determined trait because it depends on the amount of carotenoids they're able to get from their food. So this is a species where they have to be able to acquire the building blocks of these carotenoid pigments. There are certainly genes that affect foraging ability and general health, and also then have effects on feather coloration. Um, so um, when Navarra et al. Studied the um, the patterns of of androgen deposition in eggs sired by females mating with very red or less red males, what they found is when females mated with less attractive males, they put a lot more androgens in the eggs. When they mated with redder, more attractive males, the eggs had lower levels of yolk androgens. So this is interesting and it's not completely clear how to interpret it. Is it, and you also see an effect on egg position. So in both cases, females are putting more androgens in the last egg or the last couple eggs to be laid. But they're also they're also, this is if you average these bars, you'll get the data from the previous slide that females who mated to less attractive, less red, more yellow males put more androgens in to the egg. And again, the function is not completely clear because the patterns are not consistent. So egg deposition patterns are often related to male attractiveness, but in opposite directions. In house finches, that's what we just described, and then pied flycatchers, females deposit more androgens in clutches fertilized sired by less attractive males. However, in zebra finches and barn swallows, the females deposit less androgens in the clutches of less attractive males. So this is kind of interesting and weird. Are females increasing the androgens to get more direct benefits, to make chicks that beg more so that they get more feeding out of those less attractive males? Um, it's 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 interesting, because the sort of suggestion is that it depends. In some species, the males are really responsive to begging calls, and maybe in other species where they're not as responsive to begging calls, um, there isn't really a benefit of putting higher androgens. Um, but this is the sad thing is, like, this was super active, heading into like 2010, and then I wasn't able to find much more on it in the last decade. So it's one of those where I think the inconsistency of the pattern has led to being studied a little less than it would have been. But it might be a good topic for a paper to update this and see what's been found in the last 5 years. Okay, epigenetics take-homes. Most of the examples that we talked about are associated with disorders, but so were most of the earliest, first chromosomal mutations that were studied. Why? Because those are, mutations of large effect, easy to measure, easier to study, but presumably as the field matures, it'll get more into sort of adaptive traits. Some epigenetic variation is heritable. It's transmitted between generations, either by the product affecting its regulation. Those are in some of these single-celled examples, like the white and opaque forms of yeast. Some because methylation patterns are maintained in the germline. That's like licking and grooming behavior, and the vinclozolin fungicide effects. And anything that can create heritable variation should be subject to selection. So the suggestion is that, we're going to find out more about adaptive traits that are passed on not through changes in DNA sequence. Okay. I'm gonna, because the second part is much, much longer than the first, I'm gonna start on sociobiology, and then we'll take a break sometime, before or at 8. So this is talking about, a lot of the questions that, Dawkins was talking about. So we want to define altruism. Altruism, evolutionarily, is when you're acting at the fitness benefit, acting to increase the fitness of others, at a cost to yourself. And and so you want to remember kind of what, what we mean by fitness, either increase in survival or reproductive success, survival or fecundity. Both of those multiplied times each other give you reproductive success. So this is in contrast to like psychological altruism, where you're doing something not because it provides you a certain reward. So psychological altruism is kind of like an infinite loop that's hard to define or measure. This should be a little bit easier to measure. So you're increasing the survival or fecundity of another individual, and that also has to incur a cost to yourself. Now, there are in Dawkins and kind of in when you're studying this in animal behavior, there are usually four, but I'm going to add a fifth mechanism for cooperation. So the four mechanisms are mutualism, which is where the trait benefits both parties, so it's not actually altruistic. Kin selection, which we're going to spend a chunk of time on, and that is when the the evolutionary mechanism is that you're helping increase the genes that you share with relatives. And this is well explained in Dawkins. And it's pretty easy to understand. Manipulation is again when you're basically forcing or tricking someone else to behave altruistically towards you. So in this case, clearly one side benefits and the other has a cost. Reciprocity, which Dawkins talks about a bunch, and we're going to talk about some here, which is where you do something to help someone else at a cost to yourself, because later, with the expectation that later that same benefit will be returned to you. And then coercion, which can be part of either reciprocity or manipulation, where someone is forcing someone else to cooperate. But understand that coercion can also be seen in, any sort of enforcement of social norms. So we'll be talking about coercion and strong reciprocity a lot as the course continues. Okay, so kin selection is the idea that phenotypic altruism, like you're feeding someone else's offspring, so you're risking predation and getting tired and using up calories, and someone else's offspring are benefiting. But it may be genetically selfish if you're directing that helpful behavior preferentially to close kin, right? And so we want to define these two kinds of fitness: direct fitness, which is reproductive success through your own offspring and their offspring and theirs, and then grandchildren, etc. And then indirect fitness, which is how much you increase the reproductive success of related individuals. Right? So if we were thinking of we're gonna um, uh, work this out in a couple slides when we think about the relatedness coefficient of R. Okay, so um, William Hamilton wrote, I think he only had like 14 papers, but each one was like brilliant and a, you know, uh, like would start an entire field of research. Um, so one of his many contributions was the yeah, I think so. I think you used to be, I forget if I have it. They had a book of in, I could be totally wrong. It could be that there was a book of just his like most famous papers, but because now I'm curious, I'm going to take a look. Um, so let's see. Um, so the anthologies are called Narrow Roads of Geneland. I forget. Anyway, um, okay. So W.D. Hamilton developed this idea of inclusive fitness, which is your own direct fitness plus indirect fitness. Um, and so the idea is that an altruistic behavior, a behavior that help benefits others at a fitness cost to yourself, can be favored if the sum of the direct fitness loss plus the indirect fitness gain is greater than if you hadn't done that behavior. So he developed kind of a threshold. So um, RB minus C has to be greater than 0. So R is um, the relatedness towards the beneficiary. The benefit is obviously the increase in survival or fecundity, the benefit gained by the recipient, and then C is the cost to the donor. So what you can see is basically there's a discount because you share 100% of your genes with yourself, but you don't share all of your genes with even your close relatives. And so if you're increasing the survival of a close relative, maybe even your own offspring, that's going to be discounted by the relatedness. So let's first think about relatedness and how we calculate it. Um, R is the coefficient of relatedness, and it's the proportion of unique genes that you share with due to common ancestry. Um, the other way to think about it is the sort of degree of genetic similarity above that of the population mean, right? Because we share whatever it is, 99 and a half or 99.9 something percent of our genes with every other human. And so it's just what proportion of those genes that are not shared by all individuals or on average shared do we share due to common ancestry. So you could think of it as, let's say we have, mom and dad, and they have an offspring, actually, and let's let them make multiple offspring. Now, how many genes does mom and dad share with each of their offspring? Mom gave half of her genes to her own offspring, so she shares half of her genes with any of her offspring. Dad also gave half of his genes to any of his offspring. Now, this is where it gets a little bit interesting. If you want to figure out how many genes do you share with a full sibling, I'll do a little, let's change the color and ask the question. Full sibling? Well, if you were to pick any gene, if you were to take a gene from you and then ask, is it the same or different from one of the two genes that mom has at that locus, half the time it'll be the same. And then what are the odds that your sister shares the same gene through mom? Well, one half of the time it'll be the same when you go from mom to her daughter and to her other daughter. So you share a quarter of your genes through mom, so it's 1/2 squared, plus a quarter of your genes through dad. So every link in a pedigree, you share, basically you multiply by a half each time. So in that case, it's 1/4 plus 1/4, 1/2 squared plus 1/2 squared equals 1/2, right? Because each half squared. How many genes do you share with your, let's change the color. Let's say your sister has a nephew. How many genes do you share with your nephew? Well, now there's only a 1 in 2 chance that the gene that you share with your sister gets passed on to her son in this case. So it's going to equal 1/2 times 1/2 equals 1/4, which is the proportion of genes shared with a nephew. Okay, so there's a general formula that you probably now intuitively know already. And it's related to this quote that was, I forget, it's attributed to Paul Dane or someone that, would you lay down your life for your, fellow man? Of course not. But I would gladly lay down my life for that of 2 siblings, because that saves just as many genes as I share with myself. 4 nephews, 4 times a quarter is 1, or 8 cousins. And so this formula, this coefficient of relatedness R, equals the sum of 1/2 time to the raised to the power of the number of links in the genealogy. What do I mean by the sum? Well, when we did how related are you to your own sibling, right? There are 2 pathways in which you could share genes, right? I should have changed color. Right? In this case, there's the red and the blue pathway. And so in this case, it's equal to the sum of of well, let's do it like this. 1/2 squared, L is 2 for each of these, plus we'll do the sum plus the second pathway, which also has 2 links. Right? Equals 1/2. Cool. Do we actually show kin biased behavior in nature? And it's sometimes hard to really calculate the relatedness and the costs and the benefits, but there are a lot of examples of kin biased behavior. Cooperative or helpful behaviors directed preferentially towards kin. So this is a classic example that was studied by David Fenig. These are spadefoot toads, and these tadpoles are actually the same age, but they are different forms, right? So, um, so um, this is a what's called an omnivore morph, and you see it's about to eat like a little brine shrimp, Nauplius, and they, that's what they're mostly looking for, is smaller prey. And then this is the cannibal form, which is bigger, grows faster, has a much more developed like jaw morphology and musculature, and you can see it's eating another tadpole. Now, the interesting thing is these toads will have laid eggs in, temporary ponds or puddles, and usually more than one toad will lay eggs. Um, so that picture over here, they're actually the same age. So it's not just that one is going to grow into the other. These are different morphologies that they can develop into. Now, what's really interesting is whether these cannibals can preferentially consume unrelated or less related tadpoles, right? Because in a puddle with a mixture of clutch mates, full sibs or maybe half sibs, and then maybe non-sibs, it should be favored by kin selection to avoid eating your close relatives. So the next slide is actually not going to be studied in spadefoot toads, although it has been studied in spadefoot toads. This is tiger salamanders, Ambystoma tigrinum, and they also have a cannibal and an omnivore morph. And one thing that's interesting is there are some cannibals that can discriminate and some that don't. So actually, I forget, I don't know why I didn't have the slide in here for these spadefoots, but there were 2 measures. I'm going to actually just summarize the data because I remember this study. So this is David Fenig, spadefoot toad cannibals. When given a choice, preferentially consume non-siblings in choice trials and have a longer latency. It's just any tadpole with a gill. The larvae have gills. Axolotls maintain those gills into adulthood. But I mean, I think most salamanders will have a gilled larval stage. latency to cannibalize in no choice trials. So what does this mean? Um, a choice trial, I'll just write it down kind of as I'm saying it. Um, choice trial is you place an older cannibal with I think there are like 4 or 5 with small number of siblings and non-siblings. A no choice trial is exactly what it sounds like. No choice trial, place an older cannibal with only siblings or only non-siblings, and see time before cannibalism. I'm gonna see if I'm gonna try to move this guy up here. No choice trial, you place an older cannibalism with only siblings or non-siblings, and then you measure the time um, until they start. Whoa, they start to cannibalize. That's the latency. And so in spadefoot toads, they wait longer before eating relatives. They'll start eating non-relatives sooner. And if you give them a choice, they eat more of the non-relatives. So they definitely seem to be acting to, increase their genes in that they share with others by not eating them. So in tiger salamanders, this is the larval form. There are discriminators and non-discriminators. So there seems to be variation in this population. Some act like the spadefoot toads. Sure. Although if I write it down, it's saved in the recording. But sure, here's the text. So in the spadefoot toads are basically, I think, almost always discriminators. In tiger salamanders, some are discriminators like the spadefoot toads, but some of them will cannibalize indiscriminately. They will eat both relatives and non-relatives. And the idea was um, what the researchers were interested here is actually measuring the costs and benefits of being a discriminator or a non-discriminator. Because if you're a non-discriminator, you can eat more tadpoles, right? You're able to eat not just the non-relatives, but you're also able to eat your relatives, and so you're going to grow faster. If you're a non-discriminator, you might grow slower, but you get the fitness benefit of whatever offspring your siblings produce, discounted by their relatedness. And so the first thing is that there's a real benefit to your siblings when you're a discriminator. In other words, in this graph, yes, grow slower has lots of risks. You're under greater risk of predation, and you're going to be smaller, and so you're going to be able to lay fewer eggs as an adult. So slow growth definitely is costly. But so this is a little bit different. This is looking at the benefit to the siblings um, out of, I think, 5 that start in the trial. If they're housed with non-discriminators, some of them get eaten and they don't, fewer of them survive, right? So fewer of them survive because they got eaten. So there's definitely a benefit. However, that benefit is going to your siblings, and so it has to be discounted by R, by relatedness. However, the costs turn out to be low. So this is SVL stands for, I'll type it because typing is neater, snout ventral length, right? So that's a measure. So that means this is growth. So the idea was that non-discriminators should grow faster, right? And if you look at the means, yeah, there's like a tiny difference. But if you look at the standard errors include the other means, which means no difference. So it's slightly in the right direction, but that you look at that graph and you'd say there's no difference in growth rate. And then the other potential benefit is age at metamorphosis. Maybe the faster you grow, the younger you can turn into an adult and start reproducing, and that's a fitness benefit. And again, it's in the right direction. The non-discriminators have slightly younger age at metamorphosis when they turn into adults. But again, you look at the error bars and they include the other means. So no significant difference, both in the predicted direction, but not significant. So the costs seem to be much lower than the benefit that you get by having your genes increase because your siblings are still alive. So this is interesting. I mean, the funny thing is, I had a grad student who did these exact same experiments on Colorado potato beetles, but it felt wrong just showing my own data. In the potato beetle example, potato beetles are not able to recognize their kin. So they're not able to avoid eating siblings when they hatch. And they do cannibalize, they eat other eggs that are unhatched. And they get benefits from eating other eggs. But the weird thing is they don't seem to be able to reduce the kin selection costs by avoiding close relatives. And there are other species for which, like this kin discrimination behavior has been shown in a lot of species, and it's also not been seen in some other species. And I don't think we know enough to really say like why it's sometimes found and sometimes not. How are we sure that beetles don't recognize kin? Well, the problem is whenever you get a negative result, you're not exactly sure. But we gave hatchling beetles choice trials. We would do a pair of we did so many combinations. This was a grad student named Karen Colley. She would give them a choice between their own eggs and she would give the hatchling a choice between full sibs and half sibs, between full sibs and non-sibs, between full sibs and eggs taken from a different population in a different state. And in none of those trials, with choice or with no choice, and in none of those trials did she see that the hatchling prefers to eat the less related or the they always ate the first egg that they came upon. They almost always ate the first egg that they came upon. Interestingly, they are able to avoid eating, when given a choice, they prefer between a viable egg, an egg that's going to hatch, or an inviable egg, either an egg that was produced by a female that hadn't been allowed to mate, so it wasn't fertilized, or an egg that had been killed by a short period of low temperature. They prefer to eat sterile eggs. So they're able to recognize whether there's an embryo developing in the egg, and they prefer to eat eggs without viable embryos. But but they're not able to distinguish close relatives or non-relatives. Anyway, one of the classic studies of kin-directed behavior is seen in Belding's ground squirrels. So this was, I think, Paul Sherman's PhD thesis. So ground squirrels are studied for their communication because it's really fascinating. They have a whole suite of alarm calls, and the alarm calls are actually specific for different kinds of predators. They'll have one alarm call that they give for an aerial predator and a different alarm call for a snake, which ground squirrels are actually often fairly, the adults in some populations are resistant to rattlesnake venom versus a fox or a coyote. So they have really sophisticated communication system around anti-predator defense. But here's what Sherman found, what he found was that alarm calls are not given indiscriminately. In fact, they're given more often when when the um, when the surrounding squirrels, the audience, are closely related. So this was one graph from the study. This is a system ground squirrels like in ground squirrels like the prairie voles, really most small mammals. The females are philopatric, which means that the females usually, the females in a group are related to other females usually, but not to males, not to the adult males in the group. So what Sherman found was that uh, the first squirrel giving an alarm call to a predatory mammal, female squirrel, was much more likely to call when there were other females around, um, and that they were related to. And you're going to see this here. So this is kinship and differences, asymmetries, in tendencies to give alarm calls among female. Um, so Morgan asks, is this because males tend to venture out and find other groups to mate and live with? So yeah, so interestingly, both birds and mammals often have what's called sex-biased dispersal. Males and females disperse at different rates, and the general understanding is that this is like probably an adaptation to avoid inbreeding, which is costly for everything, almost everything. But for reasons that people still argue about, in birds, um, males tend to be philopatric, females are the more dispersing sex, and in mammals, it's the opposite. Females tend to be philopatric, and males are the more dispersing sex. But this leads to um, the existence, especially in long-lived social mammals, of matrilines. So you'll have mom and daughter and granddaughters in groups, and you see that in like chimps or elephants, um, whereas you don't have patrolines because the males disperse, they go off in different directions, and then they try to, become breeders in distant groups. Um, so what you see is if you're reproductive with a living daughter or granddaughter, no other living relatives, you're much more likely to give calls than if you are reproductive but without any known living relatives in the group, right? So you compare, for example, you know, what you're doing in each of these cases is comparing within these pairs. Um, and the number of observed to call are much greater than if there's a living daughter or granddaughter in the group, um, than if there isn't a living daughter or granddaughter in the group. Whereas if you took these 23, if you took these 23 calls and distributed them proportionally to the number of, reproductives with or without known relatives, that should be close to 50-50. There should be a little bit more of the ones with relatives because 27 is bigger than 24. But it's actually, much higher in that group. Um, so that's what these comparisons are, and many of them, several of them were significant, and he interpreted this as nepotism, that the calls, you're more likely to give an anti-predator call, which should be a somewhat costly behavior because it draws attention to yourself, right? So when you do an anti-predator call, the predator knows at least exactly where you are. It's benefiting everyone else that's listening. Okay, um, so one question about, so there are many, and we're going to go through some more examples of how kin selected behavior is recognized, mediated. How do they know who's kin? And one could be like a really simple rule, like whoever's near me is probably related to me because this is a family group. But it could also be through a more sophisticated, some kind of phenotype matching. And so one of the questions is, um, if phenotype matching is going on, is it due to imprinting? And so, like, um, the the behavior towards fostered nest mates should be just as strong as towards you know regular, yeah, as towards sibling nest mates. Or since mammals are really smell oriented, it was coined the armpit effect. Maybe you compare the smells, the odors of other individuals in your group to yourself, and that's what tells you how closely related they are. So you're actually trying to like match to yourself. And in that case, there's going to be more of an it. In that case, you're going to direct behaviors towards, um, you know, um, genetic siblings more than even towards like non-related individuals that you might have grown up with. And so this was studied by this was studied actually in a few cases. So I'm going to talk about Hemelipistus riamari, which is a imprinting example, and then I'm going to talk about I think it's ground squirrels. No, it might actually have been hamsters, as an example of the armpit effect. But you know what I'm going to do? It's 8, and so I am going to